So Eddie mentioned building communities, and Rob suggested earlier some interesting ideas. And I just want to emphasize the power of genomic projects to bring communities together. And I'll just show one example, which is a recent publication of Daphnia pulex. This is the water flea. This is a keystone species in the food web, sort of a canary in lots of aquatic coal mines, if you will. It's a useful both laboratory and environmental model. It was the first crustacean sequence, and it was just published. And when this project started in 2003, it was approved after discussion on the back of a cocktail napkin in Washington. There were 26 investigators who came to JGI to figure out how to do this, and the Daphnia Consortium was born. There were 9 publications on that organism that year. In 2007, the genome was released, and there were now 150 members of the Daphnia Genome Consortium, and the number of publications had risen to 18. And now that it's fully annotated, lots of people have been using it. It was just published. There were 475 investigators in the Genome Consortium, and they published 50 papers at once associated with the genome coming out. So we have tremendous power to build communities around sequencing projects. And with the commoditization of sequencing, people can think of their own experiments very easily. And one of our challenges and the community's challenge is how to figure out how to put these things together in a way that doesn't just answer your question, but helps to answer related questions by providing broadly useful data. And so in the next little bit, I'm going to step back and sort of think about how do we get from where we are today to where Eddie is leading us. And just to point out that change in genomics is evolutionary, rarely revolutionary, but it does move very quickly. And we have a strong track record of getting from here to there by scaling existing capabilities while maintaining scale, being able to do 100 times what you could do 10 times last year and 1,000 times the year after. Expanding the capabilities of what we do by adding novel capabilities, and Len gave you a nice explanation of many of the things that are at hand now. And then really trying to push the envelope by putting together bigger scale grand challenge projects that bring a broad community together, provide foundational infrastructure. And that's sort of where we'd like to go. I'm going to just show one slide to sort of show how decisions we make can really make big impacts on what we're capable of doing. So this is the output of different kinds of projects from the microbial genome program over the last few years. And if you look at the finished genomes, we're actually doing less this year. They're very expensive to produce. But as a result of the modest reduction in finishing, we're able to produce a lot more draft genomes, single-cell genomes. These are Illumina-only draft genomes, resequencing projects, RNA sequencing projects, well over 500 projects for less money than we were able to do finishing these, whatever it is, 150 base-perfect microbial genomes. So there's pluses and minuses with decisions that we make, but in general, we want to try and maximize the science that we're able to carry out. And I'm going to skip through the descriptions of the programs, as we'll have a chance to talk about that in a little bit, and jump to the actual CSP call. So, as before, we're looking for projects that are relevant to DOE missions, broadly defined. We're accepting projects that require up to a terabase of sequence. We'll ultimately decide that once projects are fully scoped, and that employ JGI's standard capabilities of de novo sequencing for fungi, bacteria, and archaea. We're not including plants this year, and I want to spend a minute saying why that is. Plants are challenging in terms of their rates of polymorphism, their complicated genome structure. We have a number in the pipeline that are really important for us to figure out and to get done. And the plant program is focused on less than our other programs on really sampling the broad diversity and is more narrowly focused on specifically the grasses. So that's why they're not included for de novo sequencing in this year. Resequencing, as Eddie pointed out earlier, is really right in our wheelhouse now. We're doing a lot of it. It works very well. Metagenome and metatranscriptome sequencing continues to be a huge growth area. We expect lots of additional growth in the program this year. RNA sequencing, both for annotation as well as de novo assembly of RNA sequence data to put together gene sets for organisms that don't have genomes completed yet. And then analysis pipelines to go along with all of these. So this is all more or less as has been the case in previous years with a significant expansion in the total size of projects that we expect. We are asking for projects that make use of JGI's still somewhat limited capacity for the things that Len talked about today. This is a real departure for us. We've been cautious about taking on projects from the broader user base, but there's really no reason not to, as long as we all recognize that what we're doing is developmental in nature. But we'll get the best projects when you put your thinking caps on and think about how you might use these capabilities as Len described them today. I won't go through this list, but I encourage you to be in contact with us if you would like to use one or more of these capabilities in your upcoming user call or upcoming proposal, so that we can make sure that we've got matching of scale, and we'd be happy to provide you with some guidance up front. Now, another word about building communities. We have historically approved a set of CSP projects, and we work out what the limits of the project are, who's going to do what it looks like, and various projects go their own separate ways. And one of the things that we'd like to try and do this year is by taking part of the allocation and putting it into specific focus areas, we like to get a collection of proposals that may potentially be synergistic. And our expectation is that we'll get those investigators within the focus areas together before the projects start to think about sharing of strategies, potential sharing of data, potential standards within the community to try and maximize the value of the data that we generate to the broader community. And in consultation with DOE, we've chosen 3 primary areas of focus this year. First being plant microbe interactions. The second being microbial emission and capture of greenhouse gases, and the metagenomics of biogeochemistry. These are areas of central importance to what DOE does, and there are areas that we believe are ripe because of the nature of the communities and the evolution of technologies that are ripe for building of these larger super communities. There's more detail about the call on our website at jgi.doe.gov, and we'll get to some questions shortly. I think we'll ultimately allocate something like 50% of this year's call toward these focus areas. So I encourage you, if this is not what you do, put in your proposals, because there will be plenty of proposals approved that are outside these areas of specific emphasis. So the timeline for the CSP call, letters of intent are due just about a month from now, end of April, 26th of April, to be exact. We will get through those in a little over a week and get you prompt notice about whether you've been approved for a full proposal. You get 6 weeks to write those proposals and send those to us by midnight on June 15th. We'll take a couple of months to review them and then review the final allocations with DOE and get your final final decisions by the 1st of September, we hope, in time for our next fiscal year, starting October 1. Another thing that we'd like to do this year that we haven't done in the past is formalize a visiting scientist program. Eddie alluded to this a few minutes ago, with the idea being that there are lots of people on sabbaticals or who have upcoming sabbaticals or who can arrange some time away from their home institution and would like to spend 3 months, 6 months, 9 months, a year at the JGI, really becoming fully engaged with JGI's activities. The idea would be that you would bring a CSP project, generate the data sets, work on the development of analysis pipelines or the application of existing pipelines to your data, get access to new technologies in a way that you can be really immersed in them, different than being at a distance, and an opportunity to engage with JGI's personnel from STEM to STERN. This could be arranged for any length of time that makes sense for you. And we're happy also to accommodate students or postdocs that might come with you to work on a project. So we'll be accepting proposals for this program pretty much on a rolling basis from now on. We'd like to have routinely 2, 3, 4, 5, 6 people at the JGI from elsewhere. So I encourage you, if you're interested, stop us in the hallway, send me an email, and there is a notice about this on our website. And your emails will come to me. And so finally, not directly related to the user call, but we do send you out a user survey every year, or investigators get a user survey. And there are several areas that are routinely come up as areas for improvement. And I just wanted to say a word about those to let you know that we are listening and we do hear you. We recognize that over the past several years, the sample submission process has gotten to be burdensome, and as projects now routinely involve dozens, sometimes hundreds of samples, our current sample submission process can be difficult. We're in the process of revamping that to make it easier. You should see that shortly. More importantly, the turnaround time for projects, both for the sequencing and for the analysis, has always been the lowest area of customer satisfaction on our survey. And not far behind it is the relative lack of transparency in finding out what's happening in your project. And we are addressing these by the development of an integrated tracking system that will follow JGI's workflows all the way from the proposal process to the final generation of data. We hope to have the initial system up and running in the fall of 2011. By knowing what's happening with samples and with your project and having it actually tracked in an automated fashion, developing reports that allow us to serve to you information about what's happening with your project should be greatly facilitated. So we have heard you, and we look forward to getting better with respect to our transparency, and we also hope that this system will make us a little more efficient, because we will be better able to track the pieces of a project that need to come together so the analysis can happen. So we're hoping that it'll increase or decrease our turnaround times for projects. So keep an eye on this, and we have heard you, and we will continue to work on these areas. And then finally, you will be receiving an email, you may have already received it, about this meeting. Please do give us your comments about this meeting and about our programs and about Eddie's strategic plan. Feel free to email us if you have additional information to convey. But please do fill out the user meeting survey. It helps us to figure out what's working and what's not working. If you like the structure of the meeting last year better than this year, or this year better than last year, let us know. So I'm going to stop there. We do have JGI's several program leads here, and I'm open to discussion about the user call further discussion about strategic plan, as well as discussion about how well we do what we do and things you'd like to see us do differently or better. No questions? You had questions for Eddie.
To some of the projects we've been doing lately. I think what's been very nice is that within the JGI we're now seeing this sort of nice collection of data which is getting more and more interlinked, which is very useful for the, I think, for the community, because this is typically something we can't do ourselves. However, of course, there's lots of other sequencing efforts going on elsewhere. And one of the problems we are having, and I mean the fungal community mostly, is that quite often the algorithms which are being used in different locations don't give exactly the same output, which you can sometimes test because the genome is in multiple places. And do you think it's going to be possible at all for the JGI to extend sort of their bioinformatics infrastructure to also incorporate these other sequence resources, so that at some point we basically can just go to the JGI and do our full genome comparison at one location rather than having to jump all over the world?
Yeah, so we're wrestling with that, not just within the fungal community, but particularly on the microbial side, where Nikos, I don't know if he's here and can speak to it, but one of the issues is whether we should re-annotate, consistently annotate all the genomes all the bacterial and archaeal genomes in GenBank, so there's a uniform standard. I think there's a lot of discussion about how to host these kinds of data, and the desire is clearly there. The question is, can you simplify the system so that you can re-annotate everything on an automated fashion and not lose quality that comes from more manual curation? And it's a balance between those two, how much manual work and how much automated work, and is the community satisfied with the eventual product that determines how that plays out. And different communities feel differently about what that should be. The plant community is very happy with, as long as there's version control, having standards that are generated based on your automated gene calls and EST data from that's empiric, and knowing that you're going to get some right and some wrong, but at least it's consistent, and when we change it, you'll know. As opposed to the fungal program at JGI, which has a model that really involves a lot of manual curation by the community, and that's really valuable to them. How does that play out in the long term? My own personal view is that we should get better at the automated annotation, because that scales. And manual curation by a community was what was required in the Jamboree era of Fugu and the first genomes that were sequenced, but it doesn't really scale now. So. But yes, we're wrestling with that. Dan, you have anything you want to add? Other questions? Well, I thank you very much for your input. As ever, this is really useful feedback for us. Please do contact us, stop us in the hallway, and give us your additional thoughts. And let's take about a 30-minute break.